Today, the topic of our class is micropropagation. At the end of this lecture, you would be able to know about what's the difference between micropropagation and plant tissue culture. You would also be able to know how can we do propagation of genetically engineered plants or transgenic plants, because transgenic in plants and micropropagation, they go side by side. They cannot prevail without the other. And also you would be able to know how to create embryos or how to create small plantlets in a relatively less amount of time vegetatively. Vegetatively means that does not involve any sexual reproduction. We know in plants this is always the case. There are anthers which contains pollens, and then they have to move to the carpel, and from the carpel they have to go down to the uh, ovary, where there is an egg which needs these pollens which act like sperms to fertilize, and then the embryo or the zygote is made. So today we will start our lecture about micropropagation. Before starting micropropagation, let's have a look about different ways of plant propagation. The first thing here is plant propagation. What is meant by plant propagation? Plant propagation means to reproduce and increase numbers. Now, if we have one plant, how can we convert this one single plant into many plants so that there will be many more? And one method is sexual reproduction, as I said, and this is through seeds. So sexual reproduction would always need seeds. So this is the non-vegetative way of plant propagation that requires seeds. As I discussed a few minutes earlier, there are structures in plants which are known as stamens, and on stamens there are anthers. And anthers are full of pollens or pollen grains, and when the anthers burst, these pollens are disseminated from the plant, from the male part of the plant, which is anther, and they reach to the carpel, which is the female part of the plant, and pollination occurs when they reach and they land on the carpel of the female part of the plant, and eventually these pollens are taken and they are used to fertilize the ovum inside the female part of the plant, and eventually it generates seeds. So both of these parts, they need dissemination, dispersal. We know wind plays a very vital role. Animals, aphids, honeybees is one of the major factors for pollination, and some other insects as well. Now, this is about plant propagation to non-vegetative. But what about vegetative, asexual cuttings? This is applicable to some plants, not to all plants. You can cut some pieces from the stems, and then you can dig them inside the ground in the land, water them, and they will grow, will develop roots and shoots, and they will turn into a whole new plant. Similarly, grafting. Grafting is one of the techniques that you can cut a branch from, let's suppose plant X, and then you would have a plant Y, a bigger tree or plant. You would make an abscess, injure the plant, and then insert this graft and somehow tie it. And in the next few days, you will see that that plant will accept that graft and it will growing. It will be like a mulberry having a graft of apricot. I am sure you have studied these things in detail in your FSC pre-medical science botany as well as in plant sciences. Then there's layers and suckers, but the most important one, which is the major part of this lecture, is micropropagation. What is micropropagation? And if you see, propagation, we discussed already, increasing the number. Micro, why do we say micro? Because we carry out this process at a size of the plant that is smaller, way smaller. Micro means smaller. In this case, it does not necessarily represent the scale 10 to the minus 6 meter. Micropropagation means small, because you would see in micropropagation, just like in tissue culture, the size of the plant will be small. We will start with small plants, and eventually we'll end up a whole bigger plant like the one that can generate from seeds. Okay, so it was discovered, or rather pioneered, by Faraday, Campion, Stewart in Cornell University in the 1950s and 1960s. That was a time when the micropropagation was really, the method was discovered and optimized. Cornell University is one of the major universities of the United States of America. And the definition is, it is the practice of rapidly multiplying stock plant material to produce many pyrogeny plants using modern tissue culture methods. Now, this is very important, this definition. If you see, we say using modern plant tissue culture, which means micropropagation is actually tissue culture. But all micropropagation that we do for plants is tissue culture, but not all tissue cultures are micropropagation. How are they different? Plant tissue cultures has several different classes. Micropropagation is one of them. Chloroplast culture is another of them. Somatic embryogenesis is another of them. Haploid production is another of them. Triploid production is another of them. So, which means to say, plant tissue culture totally relies on the totipotency of plants, and it can be further subdivided into many different sections. Of course, the techniques of all these and the sophisticated complex issues of every class of plant tissue cultures are different. But micropropagation is plant tissue culture, or is a class of plant tissue culture in which we practice the rapid multiplication of stock plant material. It is very important, and it is an in vitro technique. In vitro means, literally it means in test tubes. We perform this technique in labs under controlled conditions for genetically modified plants, for transgenic plants, and bred through conventional plant breeding methods. The plants that has been bred either by conventional plant breeding methods, you want, to increase their number. Now, micropropagation can help you a great deal in this sense, because it's relatively faster. And when you have genetically modified plants, you want to produce more plants because the important gene is already incorporated into a plant. And micropropagation in this case is really the technique that you're looking for, because of rapidly multiplying stock plant material.